The same type of thing has been shown with Coxiella burnetii, which is the causative agent of Q fever. Q fever is not that common, if at all, present in the United States, but it does occur in places like Australia and in the Middle East. It has also been shown that 10% of the people infected with dengue virus, which is a mosquito-borne virus, do not recover after a year following the acute infection. Borrelia burgdorferi, which is a tick-borne bacteria which causes Lyme disease, and that has also been shown to cause a post-infective fatigue equivalent of CFS after a year. Ross River virus and chikungunya are also mosquito-borne viruses that have been shown to cause CFS, and recently enteroviruses, which again cause an acute gastroenteritis, have also been shown to be associated with CFS. What is it about infection that can precipitate or trigger chronic fatigue syndrome? There was a longitudinal study done over several years, actually, that attempted to study acute infections with EBV, Q fever, and Ross River virus, so 3 different types of pathogens, in order to be able to identify what was it. Was it something about the pathogen, or was it something about the host that actually was causing people to be at greater risk for developing CFS? And this work was published a couple years back in the British Medical Journal, and the somewhat surprising finding was that the greatest predictor of developing CFS was the severity of the acute infection. It was independent of whether or not it was Epstein-Barr or Q fever or Ross River virus. It was almost all dependent on the severity of the initial infection. Now there are studies ongoing to try to identify what is it about that acute infection episode that might increase the risk of people developing CFS. And the investigators in Australia are actually coming up with some very important and interesting findings, which we will be reading about soon. Finally, I just want to get a little bit into some genomics and remind everybody that genes, which all of us have, and gene activity are important in helping us not only objectively diagnose CFS, but also in helping us determine appropriate treatments for CFS. So just as a reminder, our genes are our DNA, and gene expression is actually the activity of those genes, whether or not they're turned off or turned on or expressing in order to be able to make proteins. Our growth and development and how well we live can be thought of as our genes interacting with each other and in fact interacting with the environment. So we inherit our genes, and the environment that we live in affects how our genes actually express themselves. There have been a number of genomic studies now, and this is just an example of how these genomic studies can be used to our advantage. And when I say that, what I mean is they should be able to help us identify specific types of CFS, subtypes of CFS, and ultimately, so that's diagnostic, and ultimately to be able to refine the types of treatments that we have or use for CFS. This graph shows the results of a study that looked at 159 women, and this group of 159 women had both CFS, idiopathic fatigue, and controls. A number of variables, greater than 400, that were distilled down to 38 that were important. And when those 38 were used, what we were able to come up with was actually 6 different classes. 1 that was predominantly healthy, and 5 classes that were distinct from each other. And importantly, 2 of those classes were predominantly composed of people that met CFS case definition criteria by the 1994 criteria and had specific genes. This POMC and this NR3C1 are specific genes, the names of specific genes that had differences in them, making these groups distinct from the healthy non-fatigued. These other groups did not have any genes associated with them, but this one down here, this small group, did actually have another gene that distinguished it from the 5 classes. So this is important because I think it demonstrates clearly that populations or people with CFS can be distinguished from other types of CFS and each other by specific genomic or molecular profiles in combination with some clinical information. And lastly, what I wanted to show you is something that I think is very exciting in helping us understand from start to finish the process of CFS. This diagram actually comes from a proposal from one of our grantees who has recently submitted the work that we funded to the NIH for further funding. What it shows is, hold on, let me find my pointer again. There it goes. Okay, this is a picture of a brain, and the red hatched marks in here is actually space, and that's filled with fluid. And what they were able to show is that the fluid in people with CFS actually has increased or elevated lactate concentrations. Now what they tried to do with this finding is to go backwards and determine how this type of result could be. What would explain elevated lactate in people with CFS? So if we go to this cloud up here in the upper left, what you would see is that there are many possible ways to end up with lactate. And interestingly, both infection and immune dysfunction have been some of the main culprits described over the past 20 years in CFS. And these are just some relatively complicated pathways of how to get there, biochemical pathways of how to get there, in addition to mitochondrial disturbances within each cell in our body that could actually also explain elevated lactate concentrations. So my point in this showing this slide is not to confuse you, but to show you that it is in fact a complicated disease, and it is going to take a number of different types of perspectives and disciplines in order to be able to interpret this outcome. If we have elevated lactate in the brain as a marker of CFS, how then can we go backtrack, and number one, prevent it from ever happening, and number two, if it does happen, how do we objectively diagnose it and then effectively treat it?